So, this is the actual lab manual instructions for this urinalysis lab. And it's talking about urinalysis, a valuable, non-invasive procedure to evaluate function of the kidneys and other body organs. And when, I think when I first heard about urinalysis, you think like, oh, it's just people checking if you're on drugs or something, but it's way more useful than that. It's giving you all sorts of information about the functioning of your body. Anything from, like we talked about, you can check if someone seems to be having trouble with diabetes, like by having sugar showing up in your urine. You can check to see if there's all sorts of normal values for different components that would show up either in healthy people or where values, things that shouldn't show up at all, or things that show up in different amounts of pathological conditions. So you can look for all sorts of things in the urine, and it gives you a lot of good information about how the person's health is. Actually, maybe I should stop. Let me figure out what happened to my little stylus. It's so weird. When I got up to show the book, I think it knocked it off. All right, I'm going to try drawing with my finger on the screen, so it's not going to be as pretty. So when we clean catch or midstream catch, when you are going to get urine for this kind of urinalysis, you want to make sure you minimize the contamination, right? So the opening to your urethra, sitting there under your underwear, whatever you're wearing, there's all sorts of dust and whatever else and things that might be around the entrance to the urethra. So what you want to do is you want to start the stream. So you start peeing. So we'll have the beginning off screen here, but now the person's peeing and there's pee coming down. And you wait till it goes a little bit to kind of clean out the pipes, so to speak, kind of clean out the urethra and everything. And then you take your little cup and put it underneath there. And so you'll end up with this cup of urine. Maybe it's like usually fill it up about a third. You can put a little lid on it so it doesn't spill out all over there. But this is the midstream catch. This was after the flow has that's why it's called a clean catch. You use the beginning of the stream to clean out the urethra, and then you catch the urine once it's going to be more just kind of clean urine rather than any kind of contaminants that might be on the urethral opening. And again, if we were in class, we'd actually do this. If once the semester's over, or when we come back on campus, at any point you want to do your own urinalysis with the Multisticks, I'm happy to set you up with the Multisticks and little specimen containers so you can do this yourself. But for right now, you're just going to end up working with simulated data, unfortunately. But you're assuming that that's how the urine was prepared, that it was a clean catch. And then you look. There's a few things you look when you're first starting to look at it. You look at the color, right? The color tells you stuff. Clarity tells you stuff. When urine is really concentrated, it tends to be darker. Although the color can also have to do with what vitamins you've been eating. Like B vitamins can make your urine be kind of fluorescent, yellow looking. Or eating different kinds of, eating beets and stuff can make your urine look more kind of reddish because the pigments get excreted. In general, it shouldn't be too reddish if you haven't been eating anything weird, or that might be blood or something. Clarity, should be relatively clear. When people have UTIs, so often the urine looks pretty cloudy from all the bacteria and stuff in there. People who do this for a living have all sorts of cool words for the color, like amber, or like, I forget, hay or straw. There's all sorts of colors that you can use. But that's usually like the first thing you think about, is like the big picture about the urine. Like, what's the color? Is it clear? What's it like? And you think so. But then you want to get deeper. You want to get more into the chemical composition. Like, for instance, I said glucose is something you'd want to look for because that's a sign you might have diabetes. Ketones are another thing we often look for. Ketones are the metabolic they're intermediates that are formed during fat breakdown, right? So the more your body is breaking down fats for energy versus just using carbohydrates, the more ketones you'll have, right? Having ketones is not a bad thing necessarily, but it's information. What are the possibilities? If you have high levels of ketone in your urine, what are possible reasons that might have happened?
You're not consuming enough carbohydrates or fat
Yeah, no,
or
no,
sugar. Yeah.
yeah, exactly. It could be like you're on the Atkins diet, if that still exists, or these caveman Paleolithic diets or something, where you're just eating mainly meat or something, and you're not eating a lot of carbohydrates. So your body has to go to its fat stores to get the energy. So there'll be higher ketones. Or maybe you're fasting. You're not eating anything, so your body has to go to its stores of fats to make its ATP. Or it could also be related to diabetes if your cells don't have the ability to take up the sugar and have to then go into fat breakdown for ATP. Like when you have people who have diabetes mellitus, you'll often have both of these elevated. The glucose is elevated in their bloodstream because it's not getting totally reabsorbed by the tubule because it's so high in the bloodstream. But it's high in the bloodstream because it's not getting taken up by your cells. So your cells are actually having to break down fat to get the ATP they need. Um, so it used to be like when years ago, when we first started doing these labs, everything you wanted to test was its own separate test. You would have to mix the urine with these little powders and look for color changes and this and that, and look for glucose. Mix some more urine with some other little powder stuff and have an assay and could look for ketones. Um, what we have now and what we usually use in the class are these cool little things called multisticks. And a multi-stick has these little things of felt on it that change color to show you different things, and it measures lots of stuff. What I'll do is I'm going to show you a short little video just showing kind of what they look like and how they work, and then we'll talk about the different things that the multi-sticks actually measures. But first I want to give you, rather than yap about multi-sticks without you having a sense of what the heck's a multi-stick, let's look at the multi-stick in action. Do 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 do. All right, and in reality, they're getting really intense. Urine is pretty sterile in general. Like when you do blood work, you definitely want to make sure you have your gloves and maybe a gown or whatever. Um, you know, urine in general is not nearly as freaky to work with as blood. In general, it tends to be pretty sterile. Like any kind of blood work that's not your own, you really want to be paranoid about catching some fatal disease. But urine isn't quite as as weird or as dangerous that way. Right. Right. Forget about and again, right, this is a typical little thing after someone's done a clean catch. This is their urine. And you don't want it lying around too long because there will be bacteria that just start breaking it down. So you want to kind of use it pretty quick. Right? If you have urine that sits around too long and the bacteria start breaking down, you start smelling ammonia and all that. You've probably all experienced what urine smells like after it's been sitting around for a little bit. It starts getting way stinkier than it even normally is. Yeah, this guy is, yeah. Whenever you do urine analysis, you want to be really intense. Look at his, he's like, I got you. But yeah, so he was checking for clarity, color. So that is actually something that gives you information. And diabetes, sweet smelling, diabetes mellitus, gets the name. Mellitus means sweet or honey, because there's sugar. There's another type of diabetes, diabetes insipidus, which means tasteless diabetes, which is a different disease where you are not reclaiming enough water in the tubules and you pee out huge volumes of urine. So diabetes insipidus, tasteless diabetes, is different. The diabetes where you have too much sugar in your blood and therefore spilling out into the urine, diabetes mellitus, the sweet diabetes. Supposedly in the olden days, like the old, like, kind of docs would actually taste the urine a little bit to see if there's a sweetness in there, to see if as a way of diagnosing diabetes. That's actually he's checking the expiration date, or I guess in Britain, the expiry date. These go bad. Like if we have a thing of these sitting around for more than a year or so, they start discoloring on their own or start. So you do actually want to make sure, like if you have ones that are old, you might think there's something weird, but actually it's the stick that isn't working anymore. And this is actually showing you the things like glucose, bilirubin, ketones, specific gravity, pH, blood. These are leukocytes. These are all things that it's going to be able to test for. So he's dipping it in here. And then once you dip it, you're going to start a timer because the different little things take different amounts of time to develop. And basically, once you set the timer, like the first one is like at 30 seconds. 30 seconds, you can check, the color of this glucose. This is a square for glucose, and you can see, oh, if it's this kind of bluish, that means negative, no glucose. The next one up is bilirubin. Bilirubin, if it's cream colored here, oh, negative for bilirubin. Ketone here, we look here, it's also the same color as negative for ketone. If there was positive for ketone, it would be turning purple. If I was positive for glucose, I would see it starting to turn green to brown, depending on how much glucose is in the urine. So it's this color assay. You look at the square here, you compare it to the correct row on the key here, and you can tell, what's the measurement of glucose? What's the measurement of ketone? That continues up to the different stuff. So see, there's ketones, specific gravity we'll talk about in just a few moments. If there's blood, and luckily there's no blood in this person's urine, because if, again, it's kind of golden, negative is golden. If there was blood, we'd start seeing green dots. If it's whole blood or kind of green smeared out if it was hemolyzed blood. Discard. This can just go, let's forget it. All right. Wash your hands.
To complete the procedure, I would document the results of the urinalysis in the patient notes.
All right, so that is Intense Dude showing you how that works. And then this one is a little nicer. She's got kind of cool little accent.
Remove the lid from the urine sample after ensuring it is mixed.
Right, because it's just a little more, it's not as intense, a little more.
Remove a urine test strip from the container, ensuring that you do not contaminate any of the test patches. Insert the test strip fully into the urine sample and tap off the excess. Note the time and hold the test strip against the result scale on the container. Read off the results at the appropriate times by comparing the colour of the test patches against the scale on the container.
So the one thing, the way she's holding it, is a good chance these would run into each other. The other dude kind of laid it flat on the paper towel. That often's a good idea, because often the colors bleed from one into another in reality. But
record your results.
So hopefully between both of these, you can get a sense of how this works. On the module, I actually have the key here. This is what the actual key is, how you read each of these and what each of the different colors means. As we're doing this, there's going to be two ways you're going to have to analyze these multisticks. One is we actually have these kind of things here where you're going to have to, here's what the stick, it's a simulation, what the stick looks like. Here again is the thing to compare to, and you have to kind of analyze what is normal or out of normal for this particular person, assuming this stick is developed from their urine, and then you'll have to discuss it. So that will be part of this lab where it says evaluate your own urine. Normally you would dip it from your own urine, but since you can't, I just set this up. Now, if your name begins A through H, do patients A and B. If you're H through L, do patients B and C. If you're M through O, do C and D. If you're B through Z, do so part one where it says evaluate your own urine, you're basically going to look at the PDF of the appropriate patient given your name and look at the multisticks that is in that PDF, and you will then just analyze that.
Should we print out those pictures and put them in the lab notebook?
Yeah, in fact, you probably should. Yeah, absolutely. So the other thing that we often do is sediment analysis. So actually, before we get into sediment analysis, let me just briefly talk about the different things that you actually see in the urinalysis, because you are going to have to kind of describe what's going on. All right. So glucose, the first thing you can see is glucose. And again, normally glucose should be reclaimed in the PCT, that proximal convoluted tubule. So if you have glucose showing up in your urine, that's a bad sign. It might be you've just totally sugar binged and you have so much sugar in your blood at the moment that some of it's spilling into your urine. But normally it's more about like diabetes mellitus. What time? I think we got time. Let me just very briefly. So diabetes. The The type of diabetes most of you are used to is diabetes mellitus. The sweet diabetes. That's because you end up usually having sugar in your urine. This is where you have a problem with insulin working properly in your body. The other one is insipidus. Again, tasteless because the urine doesn't have sugar in it. This is something very different. This is don't kind of reclaim H2O. So you end up, remember we talked about how much filtrate is getting formed and how 99% of that is getting reclaimed. In diabetes insipidus, people pee out huge volumes. So it's a different disease, but you should know that it's a different disease. Diabetes mellitus is problems with insulin. There's two different versions. There's type 1 and type 2. And I'm drawing with my finger, so if this is looking weirder, I apologize. Type 1, sometimes called juvenile onset. Type 1 is where your islets of Langerhans in your pancreas that make insulin don't work right. It's like an autoimmune thing. Your body attacks its own insulin producing cells. So this is where you do not make insulin. And we'll talk about insulin in more detail. Insulin is basically a hormone released if you have sugar coming into your body, and it tells your cells, lots of sugar, time to take up the sugar. So if you have type 1 diabetes, you get sugar coming into your body, it starts building up in your blood, but the message to tell your cells to take that sugar up is not being released properly. Type 2 diabetes, sometimes it's called adult onset, because it tends to happen when people are older. This is where your cells do not respond to insulin. Your insulin is being produced, but your cells don't respond to insulin. So this is like a little trickier, right? If you have this juvenile onset type 1, minimally, if you know someone's sugar is going up in your bloodstream, you can give them a shot of insulin, and that will send the message to the cells to take the sugar up. This one, the cells aren't actually, even if there is insulin around, the cells aren't likely to understand the message. This one tends to be correlated with obesity or being overweight. Often when people lose weight, this gets better. And one of those ironic things, like especially like in the US with the increase in childhood obesity, it's more common now for kids to have type 2 diabetes because there's more obese kids. So this calling it adult onset isn't quite as accurate as it used to be. But again, in both of these cases, if either you're not making insulin or you're not responding to the insulin that is being made, it means that the sugar in your bloodstream is going to get to higher levels than it should because the cells aren't taking it up. And having really high levels of sugar in your blood means there's really high levels of sugar in the filtrate, which means that the PCT doesn't have the capacity to pull it all back into the bloodstream in time, and some of that sugar is spilling out into your urine. So when you are finding glucose in the urine, it's usually because there's some there's some problem with either the production or the detection of the insulin signal. And so blood has high levels of glucose, and filtrate has high levels of glucose. Bilirubin Bilirubin is, we've mentioned it actually, I said during the breakdown of red blood cells and recycling of the heme groups, you make bilirubin. It's one of the kind of breakdown products of recycling hemoglobin happening in the liver. I was saying it's also at the core of what turns like your poop brown. If the bilirubin, if something happens with your liver, like when I was in grad school, I got Hep A and my liver cells were all dying and like spilling out their guts. And there was lots of bilirubin spilled out into my system, and my eyes were basically brown. Like the whites of my eyes were yellow brown because it's just everything was turning yellow brown from this bilirubin spilling out everywhere. It's called jaundice. Jaundice is like when you have like a lot of that. But in general, if there's bilirubin showing up, that's a bad sign because that means there's some problem with the liver. Normally the bilirubin is just funneled into, becomes part of the bile and makes your poop brown at the end. Ketones, we've just talked about. Ketones are a sign that you're breaking down fats. It could be because you just are on a all meat diet and you're not eating carbohydrates, or it could be you're fasting, or it could be you have diabetes as well. Specific gravity, I'll get to in a second. Let me take a little break here. Blood, obviously, in general, there should not be blood in the urine. What's the most, especially like in a class like this, it's not uncommon for us to have the blood coming up as positive. What is likely happening if there is blood showing up in the urine in a class like this?
A UTI?
It could be a UTI. It could be a UTI, but even more common than that.
Could be menstrual blood.
Yes, usually it's very often contamination of menstrual blood. So it's like you've probably heard this or you will hear it soon enough. Like one of the classic sayings they have in like clinical stuff is like, if you hear hoofbeats, think horses, not zebras, right? So it's like, if there's something that's like there, like people often, if they've been learning about all these crazy diseases or all these things that might be going wrong, like you kind of spin out on what might this be, it's most likely the most common thing rather than something more obscure. It might be obscure, but so if you, especially in in a class where you have a lot of women of reproductive age and there's a little bit of blood in the urine sample, that's the first place you probably go. After that, maybe to a urine, a UTI. After that, maybe you talk about them, see if they've been in a bar fight like last night and got a major kidney shot or something. But anyway, so blood, pH. What should the pH of your urine be?
Neutral.
So not necessarily. Remember, I mentioned this when we started talking about the functions of the urinary system. Remember, it's doing pH balance, right?
Is it a little bit more alkaline?
Right. So it's going to depend on what your body needs to do to maintain pH balance. Depending on what your diet is, your overall blood might get a little more acidic or a little more alkaline, just depending on your diet, what kind of things you're putting into your body. And then the kidney tubules, remember we talked about the reabsorption and the secretion. Like what you need to do to maintain pH homeostasis will depend. Like let's say right now I'm a little more acidic. Then my kidneys are going to say, if I better like secrete H pluses. I got to get these H pluses out of my body to keep my pH more, in that zone it needs to be, because I'm too acidic To become less acidic, I gotta excrete H pluses. So my urine is going to be acidic because it's got more H pluses. But let's say I'm eating, especially like more vegetarian diets and stuff, maybe my body is a little more alkaline, in which case my kidneys are saying, no, what we really need to do is excrete bicarbonates because he's too alkaline. We got to get rid of these bicarbonates and bring his pH down a little bit. Which case, when you look and examine the urine is actually going to have these bicarbonates in it and it's going to look more alkaline. Right? So the pH of the urine is going to depend on whatever the kidneys need to do to preserve your pH balance. You know, with typical diets, it tends to be slightly acidic, but that's not a given. It doesn't have to be acidic. It can actually be. It could be neutral. It could be alkaline. It could be more or less acidic. It really depends on what you've been eating and what your metabolic history has been here. Proteins. In general, proteins should not be in the filtrate because they get held back by the filtration membrane, that kind of wall made by the fenestrated capillaries and the podocytes. Urobilinogen is another one of these things that can be problems from like liver disease. Nitrite. Nitrite is a metabolic byproduct of bacteria that are often found in urinary tract infections. So if you see nitrite in your urine, that is evidence that there are probably these gram-negative bacteria kind of waiting and hanging out in the wings, camped out in your urinary tract. So it's often found in parallel with leukocytes. Leukocytes are your white blood cells that are fighting infections. So if there are white blood cells in your urine, that's also a good sign that you have a UTI, because they wouldn't be there unless there was some problem going on that they needed to fight. So that is the different things that you can check. And I'll talk, let me do specific gravity quickly now. Specific gravity tells you kind of how dense, how much dissolved stuff there is in your urine. It's a ratio comparing the density of your urine to the density of water. So it's a ratio. So there's actually no units. You know, in general, in this class, you are going to lose points. In fact, you know, some of you have probably been looking over my comments on your labs. It's like, always use units, except for specific gravity, because it's a pure ratio. Density over density has no units. So it's one of the few places where you don't need to have a unit. If my urine is so dilute, let's say I've been drinking tons and tons of water, and so I'm peeing out lots and lots of water, so my urine is almost like pure water, what's the specific gravity going to be?
One.
It's going to be 1. So when specific gravity is 1 or near 1, that means your urine is almost like pure water. The more stuff that gets dissolved in it, the more dense it gets. Density is just like how heavy it is for a certain volume, right? The density, I could have like a cubic foot of lead is going to be way heavier than a cubic foot of feathers, even though they have the same volume. So if the urine has more and more of these solutes dissolved in it, it's going to be denser than just regular old water. Typical numbers, just so you know what typical numbers are, like 1.00, this would be like water. But if you have 1.01 or 1, this is starting to get saltier. By the time you're getting up to here, like 1.04, this is like really salty. Or maybe, I don't know, salt, very high osmolarity compared to normal. So even though this number looks kind of small, seeing like 1.04, this is big. This would be like you're peeing out almost pure water. This would be you're peeing out this really concentrated urine with lots of stuff dissolved in it. And again, what's the specific gravity of a typical urine? Think about the pH that I was talking about.
Depends.
It's going to depend. It's going to depend on what your kidneys need to excrete to keep a balance. Like if you've been eating a whole bunch of pretzels, you're going to have to get rid of a lot more sodium, which is going to increase the specific gravity. Or if you've been just drinking lots of water and not eating much salt, you might be peeing out something that's almost like pure water because you are getting rid of a lot of volume and very little solute, right? So this is all about the next lab, which is going to be about making your predictions about what the urine is going to look like, depending on what people have been ingesting. And you really want to think about this idea of it depends based on what they've been putting into their body. So specific gravity. The other thing that we check, whoop, didn't mean to do that. Sediment analysis. When you look at the pee in the little container, it looks pretty much just like liquid. It doesn't really look like there's anything in there other than this yellow liquid. But it turns out there is stuff. That when you're peeing, there's epithelial cells that break free of the lining of your urethra that end up in the flow. There's little crystals. I talked about big crystals becoming like kidney stones, but microscopically, there's going to be itsy bitsy crystals, uric acid crystals, phosphate crystals, this and that, which you can see if you take the urine, you put it into a centrifuge tube, then you centrifuge it super fast, and all the little stuff ends up getting smushed down to the very bottom. Maybe I shouldn't make it red because that looks like blood. This is going to be the urine sediment. And then I can take that and I can resuspend that and look at it under the microscope, and I can see the little crystals. Again, typically we do this in the lab if we're all there together. Basically, after you do the dipstick analysis, you take your urine, you put it in a centrifuge. I am not going to go, we don't have the time for this right now, but take a look. This is this guy, he's talking about some of the stuff you would find in urine sediment. All of these guys that deal with urine analysis all look like intense. I'm not sure why. Oh, it's got a fun song.
Granules and in this case,
right, this is the kind of stuff you see, like epithelial cells. They're really common, just showing up in the urine once you concentrate them down. You just concentrate your urine with a centrifuge. Look, all of you have this. This isn't just weird stuff. This is any of you, if we spun your urine down in a centrifuge and looked under the microscope, you would see epithelial cells that are sloughed off of the tubules in the urethra. these things called casts, these proteins that kind of solidify in the tubules, which give you information about the conditions in the tubules. Little crystals of different kinds, calcium carbonate crystals. Some of the crystals are normal, some of them are indicating different kinds of pathological conditions. And actually, what's normal in a dog and a human are a little different. So, oh my God, that guy's sticking his tongue out. Let's get off of that scene. Let's just get rid of it. Yeah. So take a look at this. Like in your write-up, let me look at the time. What I've asked you to do, so make sure you understand what the multisticks are. Make sure you kind of understand what sediment analysis is. Watch this guy. It's just 10 minutes of your life, and you'll get to learn a bit more about sediment analysis. And then for part 1, I just want you to, again, like I said, depending on your last name, find the correct 2 patients for your write-up, print out those 2 PDFs, and analyze what's going on with those 2 patients. You just make a little chart of what's, for each of those things, the glucose, the ketones, the bilirubin, blah, blah, blah. What is the range, and is it normal or not? And then kind of have some little discussion about what you think is going on with this person. For step 5, I say mention at least 2 things that might be found by examining urine sediment and what they might indicate, like whether it's normal or pathological. Again, you can watch this video here to get a sense of it. You can also find lots of other resources on the web that talk about urine sediment. But I do want you to specifically think about urine sediment and, again, list at least 2 things that are typical or things that are found, maybe not typical, and what they mean. For the part 2, evaluate the unknown sample. This is just kind of more of the same thing, kind of still flexing your muscles around interpreting the multisticks. In this case, there's 2 different patients. They're also called A and B, unfortunately, but they're different than in part 1. And here,
Okay,
here you're going to actually watch the actual kind of in-person, right? So watch this. And again, you want to usually have a little chart written down before you watch this. Have a little chart that just has the different categories: glucose, bilirubin, ketone, specific gravity. And then as she goes through here, write down the values. And then you're going to also do the same thing. Analyze, like, what's up with this patient. Typically we would do this like in little breakout rooms, but we unfortunately are running out of time. So you just get to do it at home. Or if you want to like FaceTime with one of your friends in the class and do this with them, that's fine as well. This usually is something we do together, but I think going over the exam, I ended up a little further behind than I wanted to be here. So this lab is pretty straightforward. It's basically looking at some of these basics about what urinalysis is, what kind of things we can look for, some of the basic techniques. But there's not much more to it than that. Are there any questions about kind of what we're doing with this one? Okay. So then I'm going to spend the next 7 minutes kind of getting you set up for the lab on Tuesday. This one's usually a really fun one. I'm bummed we're not doing it. So basically, for this one, we're going to have 4 groups. Oh, come on, goofy thing. One group that just drinks 800 mils of water and nothing else. One group that drinks 800 mils of water and also takes these salt tablets. So they have 1.5 grams of salt. One group that has very little water, just enough water basically to swallow their salt tablets, and some salt. And then group 4 has a bunch of water, no salt, but has taken 200 mg of caffeine. That's about like a couple of cups of coffee. That's a no-dose or a Vivarin. And basically, you have these 4 groups. In fact, let me now go into my... So we have our independent variables, which we just talked about. There's going to be like H2O intake. How much water are they drinking? Right? It could be anywhere from 80 mils to 800 mils. There's going to be salt intake. Some people are taking salt tablets. Some people are not. Then we have caffeine intake. Some people are having caffeine and some are not. So these are the things we are messing with. We're changing how much water they're taking in, how much salt they're taking in, and how much caffeine they're taking in. The dependent variables, the things that we're going to measure, every half an hour we're going to look at the volume of their urine, how much are they peeing? And this is not going to be a clean catch. This is just going to be the full amount they're doing. Sodium chloride concentration of the urine. How salty is their urine? And then the specific gravity as well. In general, specific gravity and sodium chloride concentration are going to be related. The more sodium chloride dissolved in there, the higher the specific gravity. And what you need to do is come up with a hypothesis about volume, sodium chloride concentration, and specific gravity for each of those 4 groups. Ready? Like group 1, 800 mils of water and no salt. What are you going to expect? Like when you come up with your hypotheses, really just think about osmolarity homeostasis. So remember, homeostasis is going to be at the core of everything. Don't get super fancy about anything. Don't worry about this intricate interplay of things in a nephron. Think about the big picture. In the big picture, what is the kidney doing, and what is probably going to end up being reflected in looking at the urine coming out of this person. Basically, when at time equals 0, they're going to drink their water and salt and whatever else. And then t equals 30 minutes. We're going to look at t equals 60 minutes. We're going to look, etc. We're going to see how does the urine, what's happening to the urine as time goes on after they've done their initial taking in the water and salt and stuff. So if you have somebody who drank a whole bunch of water, that's like a big gulp of water, and didn't take any salt, what are we going to expect in terms of their urine as we watch over the next 2 hours?
Volume will increase, sodium will decrease, and specific gravity will decrease.
Right, exactly. Right. If we want to maintain our balance, right? If we want to maintain water balance, and I'm drinking a buttload of water, I got to let go of a buttload of water in order to keep my balance. So if I just drank almost a liter of water, I'm going to have to pee out most of that to keep my osmolarity the same. Right? That's the whole point of the kidneys, that water balance. So, and that means there's going to be a large volume of urine. That's how the water is leaving my body. It's in my urine. If I'm not eating any salt, then my body is probably going to want to hold on to what it's got, assuming it's already in balance. So I'm probably not going to have a lot of salt showing up in my urine, because in order to maintain my osmolarity homeostasis, my electrolyte homeostasis, I'm going to hold on to my salt. So I won't see it in the urine because I'm holding it into my body, reabsorbing it. Which, as Dana was saying, is also going to mean that the specific gravity is probably going to be low if there's not a lot of salt in the urine. And what about group 3? Or is it group 3? There was only 80 mils of H2O, like really dinky amount of water. It's really like a little Dixie cup, and a bunch of salt. What are we going to expect in somebody like this?
The volume would decrease.
Yeah, so if I am not putting any water, that means my body has to hold on to its water, or else I'm going to start getting more dehydrated. So I'm probably going to have a much lower volume of urine because my body's reclaiming its water rather than letting it go to maintain balance. However, if I'm taking an extra salt, I'm probably okay letting go of more salt to keep my balance. And in fact, we'll see there's more going on. If you have less volume, that means you're going to have less, since concentration is actually a ratio of like salt per volume, having less volume also means probably higher concentration for the same amount of stuff dissolved even. That makes sense. Right? If I have a teaspoon of salt in a huge beer mug and a teaspoon of salt in a tiny little shot glass, it's the same amount of salt, but much lower concentration in the beer mug. So for each of the 4 groups, predict, what and why. And again, the why is usually going to be about like the balance. The volume is going up, so I need to release water to keep the volume back down. What and why for our volume, sodium chloride concentration, and specific gravity of our urine. Remember, we're not going to know exact amounts, right? We don't have enough data for that. So it's going to be relative. Which one is going to be like, what's going to be higher than the other or lower than the other, or maybe the lowest or the highest of the groups. So it's all going to be relative. Don't outguess yourself trying to get too fancy. It's kind of relative amounts. So if you go into here, it says, so here's a little background thing you can look at. I mean, we're at 12:30, so I'm going to just let you see this on your own. It's basically a little 2 minutes and 30 seconds where Becky is kind of showing you what things look like. But here it says, when you're writing your hypotheses, think about essential roles of the kidney, in particular water balance and electrolyte balance. So the whole point of this is thinking about what are your kidneys doing in the big picture, and how is that going to be reflected in the composition and volume of the urine. This before Tuesday, you should have this done. Again, right here is just to submit PDF of kidney regulation of osmolarity pre-lab. That's a mouthful. Kidney regulation of osmolarity lab pre-lab. And if you're thinking like, oh my God, another pre-lab, remember after this, there's only one more pre-lab, and then we'll never have another lab, at least for this class again.